The field's potential for basic science appealed to him, but Luthart was equally captivated by its promise to transform not merely the medical field, but the very experience of being human. In essence, Luthart saw in neuroprosthetics something unprecedented in roughly 200,000 years of human history, an entirely new pathway for the brain to manifest its intentions. By wiring the brain directly to a computer, researchers might unyoke thought from its traditional confinement to the brain and body, bypassing a biological dictate that has held sway since the first invertebrates formed loose nerve networks to move about. It seemed like an inviolate rule of biological evolution. Organisms would rely on a class of interconnected information-carrying cells known as neurons to direct muscles, their sole means of interacting with the environment. But with monkeys taking mental control of cursors and rats moving feeding levers, these early researchers were tilting at a radical new vision of how the brain might interact with the physical world. By grafting the brain directly onto a computer, they were clearing the way to extend the body's biological nervous system of dendrites and axons into a digital realm of zeros and ones. As Luthart saw it, neurally controlled appendages were just the beginning. The increasing digitization of the environment meant the brain could someday interact wirelessly with a host of surrounding technologies. Everything from smartphones and laptops to climate controls and lighting. Maybe for no other reason than I think it's amazingly cool, he said, but that's cracking something. After all, we only need to move a mouse in 3 directions, up, side to side, and click to gain control of a computer. That's merely 3 degrees of freedom, a feat some researchers have already demonstrated. Why couldn't a neural augmented communicate with a smartphone or wearable tech, granting users not only the internet, but also to a communication hub for surrounding technologies? It was a thrilling prospect and one that opened a broad challenge to time-honored notions of evolution, biological order, and necessity. Our best estimates place the age of the world at roughly 4.5 billion years old. The fossil record indicates that simple one-celled organisms, ancestors of today's bacteria, ruled the Earth until about 1 billion years ago, when these life forms began to mutate, ushering in a generation of multicellular organisms, not unlike today's sponges. The first animals crawled from the sea around 500 million years later, and it took another 500 million years for the hominins to arise, which occurred a mere 7 years ago. Through it all, genes ruled. Counterintuitive though it may be, a gene's objective is not the survival of a particular animal or species. A gene's primary aim is its own replication. The survival of the animal it happens to construct, or, as the evolutionary biologist Richard Dawkins would have it, the genetic vehicle, is incidental, important only in so much as it provides a successful means for genes to survive and replicate. It's a distinction that is easily overlooked, but in genetic terms, an animal is little more than a temporary structure genes build to ensure their replication. In this sense, it's largely irrelevant whether a gene survives in a flatworm, a giraffe, or a human. What matters is that collections of genes combine to create biological vehicles, organisms that are successful in specific habitats. Of course, genes optimize a biological vehicle through the fumbling process of genetic mutation, the evolutionary mechanism that has governed life for the past 3.5 billion years. At least it was until 160,000 to 200,000 years ago, when our species arose from the hominid line to offer what some evolutionary biologists describe as an alternative to the genetic monopoly, or culture. Just as biological vehicle protects the genes it houses, so too does the culture allow groups of humans to survive by forming what the evolutionary biologist Mark Pagel calls Cultural survival vehicles. Pagel argues that, like the protective plates of an armadillo or kinetic muscle and bone architecture of a cheetah, culture enmeshes us. It empowers groups of humans with shared language, common identity, and inherited knowledge base. It is the crowning adaptation of our species and wraps us in a protective mantle that defines us, enabling our survival in the sense culture is not so different from a beehive or an ant colony, a collective activity that functions almost as an organism self, protecting individual members that would quickly perish on their own. Like social insects, humans exist almost exclusively in organized groups. We may not be able to draw physical boundaries around a culture, but our cultures, no less than a body for genes or hive for bees, that have been critical for our success. The difference, of course, is that while social insects have defined activities, individual members of a culture are relatively free to play a host of roles. What's more, culture is cumulative, meaning that ideas, the tempering of steel, say, or invention of penicillin can be transmitted not only from one person to another, but also from one generation to the next. As Pagel writes in Wired for Culture, it is hard to overemphasize its evolutionary importance. Our invention of culture created an entirely new sphere of involving entities. Human acquired the ability to learn from others and to copy, imitate, and improve upon their actions. This meant that elements of culture themselves, ideas, languages, beliefs, songs, art, technologies, could act like genes, capable of being transmitted to others and reproduced. But unlike genes, these elements of culture could jump directly from one mind to another, shortcutting the normal genetic routes of transmission. And so our cultures came to define a second great system of inheritance, able to transmit knowledge down the generations. For humans, then, a shared culture granted its members access to a vast store of information, technologies, wisdom, and good luck. Having culture is why we watch 3D television and build soaring cathedrals, while our close genetic relatives, the chimpanzees, sit in the forest as they have millions of years of cracking the same old nuts with the same old stones. Pagel argues that our genes themselves have evolved but not to inhabit any one physical habitat, but rather to inhabit the most important environment for our species, the social environment. Our genes have produced a neural architecture that creates the mind, a mind that is flexible enough to embrace any cultural environment it happens to be born into. This behavioral flexibility enables us to acquire that Pagel calls social learning. Whereas other animals can learn new behaviors, humans can not only learn a new behavior, but also understand the utility of what we've learned. We can translate it into a new environment, passing novel behaviors on to others and even improving on them. It's the reason we now inhabit all corners of the globe. While other species rely on genetic adaptation to explore new environments, humans need only tap our vast cultural resources to take on novel traits. In earlier times, this might have meant learning that fletching makes a spear fly straight or that iron tempered with carbon forms a stronger metal. More recently, antibiotics have enabled us to ward off deadly bacteria. Advances in genetic laws created new breeds of disease-resistant crops, and computers have enabled us to collect and analyze unthinkably large troves of data. These are just a few of the millions of ideas that have moved through the human population, enabling us not only to take on adaptive traits with lightning-like speed, but also to thrive in environments we once found hostile. It might have taken the modern polar bear eons to develop the layers of fur and fat that enable it to live in Arctic climates. By contrast, we can adopt the traits necessary for Arctic living by tapping a veritable genome of ideas, everything from leather tanning and high-tech fabrics to extracting and refining fossil fuels and internal combustion engines. Instead of adapting to the demands of any one physical environment, our genes have evolved to use the new social environment of human society to further their survival and reproduction, writes Pagel. These are adaptations that have wired our minds and bodies for culture. Through culture, we have in some essential way transcended nature. We have become the drivers of our own evolution, and though we may not foresee the cumulative effects of certain ideas, we are nevertheless able to introduce new ideas or mutations into our cultural survival vehicles, our hives. Ideas and brains are new information carriers that are shaping evolution, said Luthart. There's a whole new organism that is created that we're not aware of. Humans and the cultural and social infrastructure are all part of this organism. It's self-maintained and I would argue self-aware beyond our potential capacity to understand its awareness. Call it Homo socialis, and just as the biological nervous system becomes more developed with neuroprosthetics, you're seeing a new nervous system develop. You create a much more integrated organism, just as the printing press created a more integrated organism, just as the telephones created a more integrated organism, just as the web, Twitter, all of these things. Culture might have freed us from some of the evolution's genetic fatalism, but there is no disputing that in many ways we remain beholden to our biology. We still get old, and bearing birth defects, we are each born with a pair of arms and legs. No matter our cultural achievements, we have historically remained bound by genetic evolution and our neuromuscular mode of acting in the world. But it was precisely this evolutionary tenet that earlier BCI researchers were threatened to disrupt. This was not a trait that had been handed down through generations. Rather, by splicing a digital nervous system onto the brain, they were promising to bypass the slow, hit-or-miss modulations of genetic evolution, swapping it out with a deliberate, non-biological innovation. It's an engaging notion, said Luthart. Your ability to interact with the world is no longer constrained by the length of your arms. The image of the cyborgs has inhabited a contentious plot of cultural real estate ever since Manfred Kleins and Nathan Klein first coined the term in their 1960 paper audaciously titled Cyborgs in Space. The Soviets had humbled the Americans 3 years earlier by launching the unmanned Sputnik 1 into orbit. The Americans countered 4 months later when they hurled Explorer 1 into space in January 1958. Yuri Gagarin's history-making flight was still a few years off, and although neither country had launched a man into space, it didn't stop futurists from imagining the bodily rigors of interstellar travel. Klein, who had been awarded a prestigious Lasker Award for medical research, ran the Dynamic Stimulation Laboratory at Rockland State Hospital in New York, where he had recently hired the young polymath Manfred Kleins as his chief research scientist. Kleins had grown up in Australia and attended Juilliard School of Music, where he'd become a concert-level pianist. He studied the psychology of music at Princeton University, where he befriended Albert Einstein. By 1960, however, Kleins was ensconced in the lab at Rockland, which the New York Times then described as resembling the back room of a radio-television repair shop. It was in that shop that Kleins developed one of his most lasting inventions, the Computer of Average Transients, or CAT, a noise-canceling device for electrical brain research. By quieting some of the brain's background noise, Klein's device enabled researchers to isolate the neuronal reaction to a specific stimulus, say the color blue or the sound of a train whistle. After repeating the train whistle for a few times, scientists could average the neural response to determine the brain represented or experienced. Specific colors or sounds. Klein's machine established a direct and articulate communication system between the brain and the outside world. The early device measured sensory input, a train whistle, the color blue, and correlated it with a measured neural response. The neural reaction evoked by a specific color or sound. The beauty of the CAT system was that it bypassed a person's conscious experience. Instead of asking a person about his experience of the color blue, the CAT system enabled researchers to observe directly how the brain responded to stimuli. Of course, the meaning and mechanics of that neural response remained a mystery. Nevertheless, Klein's machine gave researchers a powerful functional tool. With the CAT system, they no longer needed to rely on the muddying vagaries of subjective experience. They could observe the brain as it represented the outside world to itself. But the CAT system did something less tangible, too. Klein's unveiled his machine in 1960, the same year he and Klein published Cyborgs and Space. And the machine served as an early example of the sort of symbiotic man-machine relationship their paper articulated. The first moon landing was nearly a decade away, and exploration of deep space remained more in the realm of science fiction than actionable science. But the scientists couldn't resist the draw of interstellar travel, speculating what it would take for space travel involving flights not of days, months, or years, but possibly of several thousand years, which they warned would eventually be hard realities. The researchers argue that astronauts would have little chance of surviving deep space if spaceships simply mimic the Earth's environment. We place ourselves in the same position as a fish taking a small quantity of water along with him to live on land. The bubble all too easily burst. They proposed instead as astronauts be biologically adapted to survive an extraterrestrial environment. They called this adapted human a cyborg, which they defined as an organism that incorporates exogenous components extending the self-regulatory control function of the organism in order to adapt its new environments. In essence, the scientists were proposing a form of elective, non-genetic adaptation. They argued that space travel was not only a technological challenge, but also a spiritual challenge, one that invites man to take an active part in his own biological evolution. In the past, evolution brought about the altering of bodily functions to suit different environments. They wrote in the September 1960 issue of Astronautics. Starting as of now, it will be possible to achieve this to some degree without alteration of heredity by suitable biochemical, physiological, and electronical modifications of man's existing modus vivendi. The original version of Cyborg was decidedly tame compared with the replicants Harrison Ford hunted down in Blade Runner or Arnold Schwarzenegger's killing machine in The Terminator. Rather, the term's creators believe that instead of giving an organism superpowers, a cyborg's technology should be biologically integrated to maintain the astronaut's basic functioning, helping him breathe, process fluids, and maintaining muscle tone in the oxygen-deprived and weightless environment of space. Instead of creating a new breed of superhuman astronauts who consciously controlled their cyborgs' technologies, they envisioned a union between man and machine that, like the CAT system, bypassed the astronaut's conscious mind. The technologies would interact directly with the body, regulating, transforming, and performing specific bodily functions. Some of these technologies would induce hypothermia and slow the metabolism so astronauts wouldn't need as much food on board. So would act as respiratory systems that did not rely on breathing, while others would reduce and recycle bodily waste before intravenously reintroducing it as hydrating fluid. This self-regulation must function without the benefit of consciousness in order to cooperate with the body's own autonomous homeostatic controls, they wrote, undaunted by the audacity of their proposal. If man in space, in addition to flying his vehicle, must continuously be checking on things and making adjustments merely in order to keep himself alive, he becomes a slave to the machine. The purpose of the cyborg, as well as his own homeostatic systems, is to provide an organizational system in which such robot-like problems are taken care of automatically and unconsciously, leaving man free to explore, to create, to think, and to feel. It was a far cry from the sleek flowers of Robert Downey Jr.'s Iron Man. Rather, the scientist had articulated a critical insight about our relationship to technology. It is through technology that we fully realize our humanity. Mid-20th century neuroprosthesis like cochlear implants and deep brain stimulators have already ushered in a sort of cyborg envisioned by Klein and Klimes. And while neuroprosthetics working with robot arms and cursors offered a tantalizing glimpse of what the field could achieve, it's based on what Luthart considered a conservative view of the body. BCI might offer a host of new sensory experiences simulating the brain and so that users are better able to retain information or endowing people with so-called sixth senses, enabling them to perceive portions of the visual spectrum our biological eyes cannot. Could a BCI transmit our thoughts directly to another person? How about our feelings, both emotional and sensory? Neural augment could be just as effective in an online network as with surrounding technologies. But what happens when networks become amalgamations of man and machine? Cyborg webs where machines and humans work in tandem. I don't even like the term cyborgs anymore, Luthart said. The extension of our personas is no longer just the fact that we attach a machine to ourselves. It's how we network, how we can perceive have a presence beyond our physical bodies. With neuroprosthesis, Luthart envisioned more than merely manipulating the immediate environment. So how does that change us, he asked? As we expand our capabilities, we potentially expand our limitations and vulnerabilities. At some point, we are going to be able to be hacked. The conversation between neurons and machines is a complex one and conducted in many languages. Ions and hertz and amplitudes, yes, but also palladium and proteins and glia. Luthart knew we needed a better understanding of bioengineering if we wanted a legitimate shot at his realization, his prosthetic dreams. He would need access to a lab, so when he arrived at resident at Washington University, he set up a meeting with Frank Yin, who chaired the school's biomedical engineering department. Then in his late 50s, Yin had arrived in St. Louis from Johns Hopkins University. He was a founding fellow of the American Institute of Medical and Bioengineering. He was a fellow of the American Society of Mechanical Engineers, and he served as the president of the Biomedical Engineering Society. He was also deeply skeptical of his young surgical resident with thick dark hair, outsized ambition, no engineering experience, and fascination with a field that sounded as much like science fiction as it did science. Yen's graying eyebrow rose even higher when Luthart told him he'd studied theology in college and was now angling to spend a research year in biomechanical engineering. Let me get this straight. You've never taken an engineering class and you studied theology. Luthart recalled Yen saying. He was exceptionally dubious about my presence. After a few tense moments, Yen told Linhart not to talk to a newly hired researcher who could probably use a few bodies to warm up his lab. The new scientist was Dan Moran, a turkey-hunting, tobacco-chewing, steak-eating farm boy from small-town Wisconsin, who just happened to be coming off a 6-year postdoc as one of the country's premier neurophysiology labs. Moran was a pure engineer, both in training and in temperament. At 15, he had completely reconditioned his first car, a 1966 Chevy Impala Super Sport 327. He gave the car new rings and pistons. He installed double spring lifters in the rear so it crouched like a bull ready to charge. But Moran's fascination with machines extended beyond mere horsepower. He'd spent his adolescence steeped in the prime-time adventures of Steve Austin, better known as television's $6 Million Man, an astronaut who was rebuilt by government surgeons after a devastating crash. Doctors implanted Austin with a bionic eye that had an onboard zoom lens. Bionic legs meant Austin could run at 60 miles per hour, chase down cars, and kick through steel doors. His bionic arm had metal crushing strength. It even came with a Geiger counter because, well, as the show's narrator said during the opening credits, "Gentlemen, we can rebuild him. We have the technology. We have the capability to make the world's first bionic man. Better than he was before. Better, stronger, faster." The show ran only for 4 years, but its cyborg promise left a deep impression on Moran, who collected the action figures and who, to this day, will occasionally sneak an online rerun or two. It was his sophomore year, and Moran, who played catcher, was riding the bench with a broken arm. He watched from the dugout as his friend, churning hard off third, dove headfirst to beat the tag. It was a freak accident. His friend collided with the catcher's shin guard, snapping his neck like that, Moran recalled, smacking his hands together. He's been in a wheelchair ever since. His head was paralyzed from the neck down. As Moran continued to restore his beloved Chevy, he realized he would spend the rest of his life working to transform his primetime fantasy into a 21st century reality. Moran would make his own $6 Million Man. With the $6 Million Man, it was the superhuman strength, right? You can go beyond human performance because you're a superhero. But when my friend broke his neck, it was no longer about being a superhero. It was about getting back to normal, he said. I realized this was a TV show, that this stuff hadn't been invented yet, but that didn't mean we couldn't invent it. To that end, Moran spent 5 years as a graduate student at Arizona State University writing equations to reproduce the musculoskeletal biomechanics of bipedal motion, also known as walking. A wildly complex enterprise for anyone who has ever had to think about it. Movement for Moran was a question of torque, joint angles, force, and velocity. There was just one problem. I didn't know anything about the brain, he said. I'd never recorded a neuron. I'd never even seen a monkey. If Moran wanted to recreate the true nature of bodily motion, he would have to go to its source. He would have to study the brain. It was about this time that Moran met Andrew Schwartz, who was then making a name for himself by charting how small neural populations could be correlated to arm gestures in monkeys. I heard Andy Schwartz talk, Moran recalled, and I was like, oh, that's it? That's my guy? Moran did his postdoc in San Diego, where under Schwartz's tutelage, he learned to open the skulls of monkeys and slide hair-thin electrodes into the motor cortex. He learned to keep the wire snug against a neuron and to record its activity. He listened to their crackle and hiss, developing a series of algorithms that could transform their raw activity into a coherent set of motor commands. When he arrived in St. Louis, Moran's tools of choice were still the slender, penetrating microelectrodes he used with Schwartz. But Moran was a pragmatist. He hewed to the same get-her-done ethos that had carried him through a boyhood of trapping game and customizing cars. Microelectrodes might be great for basic research. They sidled up to an individual neuron and delivered exquisite cellular recordings. But their delicate wires were brittle. They also provoked an immune response encapsulating the electrode. There was no getting around it. Penetrating electrodes were simply too fickle for the sort of day in, day out, $6 Million Man type brain implant Moran aimed to develop. I can't go to a person and say, I'm going to give you fabulous control, but it's only going to last for a year, he said. Who's going to have brain surgery like that? The joke about Moran is that while many people don't like to micromanage, Moran doesn't even like to macromanage. Chalk it up to a native strain of stoicism forged on the fields of Wisconsin, but Moran is what Luthart called a classic 10%er. Say one of Moran's monkey studies winds up in the pages of Science. His baseline mood improves about 10%. Now say hydrogel he's working on to prevent electrode encapsulation is as effective as a jar of Vaseline. Again, the needle moves about 10%. Moran doesn't roll easily. He's given to wearing short-sleeved polo shirts, crispy blue jeans, and Birkenstocks or some other sensible brown shoe. By the time Moran and Luthart met in 2005, the question of encapsulation seemed like one of the most urgent in all neuroprosthetics. Moran had spent the better part of the decade in planning penetrating microelectrodes into the brains of monkeys, and with only one exception, a storied macaque known as Mojo. He had watched with dismay as his recording quality inevitably deteriorated. As Moran saw it, he had two choices: either develop a novel technique to make his microelectrodes more robust, or invent a whole new way of coaxing information from the brain. Neurosurgeons had long used a technique known as an electrocardiography, or ECOG, to detect seizures focus. More recently, a handful of scientists had used the technique, placing electrode grids directly on the surface of the brain as a research tool to record neural activity, but these were hardly brain-computer interfaces. They weren't trying to translate the information into motor commands. So Moran had carried on, sinking his wires into the brain of rats as he built his new monkeys lab in St. Louis. He was also experimenting with biomaterials, trying to develop everything from a Teflon-like substance to prevent proteins from attaching to electrodes to anti-inflammatories that would suppress the brain's immune response. But the research wasn't going well. As in San Diego, Moran would get splendid single-unit recordings initially, only to watch them fade over time. Moran's mood needle moved about 7% whenever he thought about encapsulation, something he did each time he spoke with the potential lab member. But Luthart was different. Like Moran, the young resident wanted to understand the language of the brain. But Luthart came from clinical perspective, and while he may not have had Moran's research background, he had something else: human epilepsy patients. Eric was like, I've got these human patients with grids on their brains, Moran recalled. I was like, really? You've got those? It didn't dawn on me that every big surgical place would have these things. The idea soon emerged that they could use these temporary neural implants to decode the brain's intention. They could use it to control machines. Because the grid went directly onto the brain, it would have greater fidelity than an EEG. By the same virtue, it didn't penetrate the brain, so it might sidestep the problem of encapsulation. A whole new paradigm was opening before him. Okay, Moran thought to himself, do you want to keep hoping that people will someday solve the single unit problem? Or do you want to get something into patients in your lifetime? The pure scientist goes with option A, but the engineer goes with option B. The neurosurgeon goes with option B. The option B, what it was, as they soon agreed that Luthart would spend the next year working along two sides of research. The first, a sort of safety study likely to bring publishable results, would use rats to investigate the brain's response to penetrating microelectrodes. But the second was pure speculation. They would use ECOG to try to produce an unprecedented brain-computer interface. Unlike with an EEG, which attaches sensors to the scalp, their electrodes would slide below the skull. Unlike microelectrodes that pierce the brain, their platinum sensors would ride above the cortex, leaving it largely undisturbed. Better yet, ECOG would give them a distinct edge over the single-unit researchers who then dominated the field. Those researchers had spent their careers plunging electrodes into the brains of rats and monkeys, but Luthart Moran would be able to get their electrodes on a substance that remains remarkably rare in neuroscience: live human neurons. By piggybacking their research onto epilepsy patients, they would also avoid the thickest of the federal regulations that had prevented invasive brain researchers from working in humans. The FDA had already approved ECOG grids for human use, Now it was simply a matter of getting epilepsy patients who were already planning to be implanted to sign off on the experiments. Agreements that would take other researchers months, if not years, and back and forth with the FDA were simply unnecessary. Still, no one had ever used ECOG for much of more than a seizure monitoring, and hospital administrators prohibit the researchers from simply running a splitter off the seizure monitor electrodes. That would give that would have been a simple fix, but the hospital needed assurance that his new-fangled research track wouldn't interfere with patient care. If Bluthart and Moran were going to tap the clinical monitoring system, they'd have to do it separately, waiting until the clinical side had the information it needed. That's when things got complicated. Electrodes record brain waves as an analog signal. Both clinicians and researchers must digitize those signals for computer analysis. Normally, this wouldn't be a problem, just as MP3 can be copied repeatedly. Luthart and Moran could have simply borrowed the digital signals neurologists used to study seizure focus. The problem was that clinical system digitized the signals in a way that made it impossible for Moran and Luthart to break them down for decoding. They would need a workaround, a device to digitize signals on their own. They would need, in other words, to redigitize the digital signals, rendering them into a readable format. As they tossed around various ideas for jacking into the system, they realized they could use an old brain printer, that kind of neurologists once used to print brain waves on reams of paper. The printer wouldn't know where the signals were coming from. Why not feed the network's digital signals directly into the printer? It wouldn't matter where the signals came from. The printer would read them as if they were coming straight from the brain itself. Luthart, meanwhile, spent the next few months collecting off-the-shelf electronics as he built a second digital converter. The idea was to feed the network's digital signals into the analog printer and then redigitize the signals with Luthart's converter. It was the neural equivalent of recording a live performance on an LP, converting the album to an MP3, recording that MP3 to a cassette tape, and then redigitizing the cassette tape as an MP3 file. In other words, it was a total kludge. But if the system was inelegant in theory, it was downright ugly in execution. When a patient comes in for epilepsy monitoring, the analog signals come off the patient's brain, where the electrodes funnel them into an amplifier and digital converter. Fiber optic network then ferries them into the epilepsy monitoring unit down the hall. Because the hospital's higher ups wanted to ensure the research didn't interfere with patient care, Moran and Luthart had to patch into the system after the signals arrived at the monitoring unit. As Luthart and Moran strategized, lining up research subjects, coordinating with Ogeman to implant the grids, and building their Rube Goldberg signal converters. And they contacted Jerwin Schock, a young researcher scientist at the Wadsworth Center in Albany, New York. Some people talk with their hands, others talk with their arms. Then there are people like Schock who simply brutalize the air, chopping and swiping and pushing. Known as Jerb to everyone in the field, Schock played semi-professional football in his native Austria. Now he has the habit of standing with rounded shoulders in that slouchy, muscular way of athletes who have proven themselves on the field, but now must go about the mundane task of daily living. Schock's forehead is low. It's framed by a careful muss of brown hair and deep crease that traverses the bridge of his nose. He cuts an unlikely figure for a scientist, with small blue eyes and fleshy cheeks. But if Luthart and Moran took circuitous routes to neuroprosthetics, Schock hit it sideways. Growing up in the tech backwater of rural Austria in the 1980s, Schock became a sort of proto-hacker at the age of 12, when he began toting his Commodore VC-20 in a backpack. He spent years writing software code, building early computer models, and bespoke information systems. By the time Luthard Moran contacted him, however, Schock had parlayed his computer know-how into a job as a research scientist in a prominent EEG lab at Wadsworth Center in New York. Like Moran and Luthard, Schock was looking for a more reliable brain-machine interface. But while Moran cut his teeth on penetrating electrodes in San Diego, Schock had learned at the feet of Jonathan Wolpaw, one of the country's foremost EEG researchers. Still, Schock was no neuroscientist. He'd never even taken a cell biology class, absorbing what he knew instead of his work in Wolpaw's lab. What Schock did have, however, was a deep knowledge of computer systems and programming, which made him irreplaceable in Wolpaw's lab when he designed BCI 2000, a software program that converts brain signals into some form of motor output, moving a cursor on a screen or manipulating a robot arm. The software codified the relationship between brain and machine, allowing researchers to change the parameters of both the brain waves they fed into the software and the action those brain waves produced. The software could also track external stimulation in time, having a research subject watch a cursor move to the right, for example, and correlated the visual stimulation with specific brainwave activity. It was just sort of thing Moran and Luthard would need. But Schock had his doubts. For one thing, no one had ever used ECOG for a brain-computer interface. Schock also came from the world of EEG, where researchers laboriously trained research subjects to achieve even modest two-dimensional control. The signals were easily corrupted, not merely by surface muscle activity, but also something by ubiquitous as ambient electricity, which could make the signals go haywire. ECOG would use the same techniques as EEG to pull information off the brain. But Luthard and Moran would have to conduct their experiments in a hospital room that positively crackled with electronics. How could their warmed over, redigitized signals not end up corrupted? There were too many variables. Finally, the payoff might not be so great. We don't even know if this will be any difference in performance, said Luthard, imitating Schock in his best Schwarzenegger accent. We don't know. Unlike an EEG, where inch-thick barrier of flesh and bone acts as natural low-pass filter that absorbs higher neural frequencies, ECOG had no such limitation. This makes it a powerful diagnostic tool when hunting for the epileptic fountainhead. Still, anything much above 70 Hz is thought to carry very little useful information for seizure detection. So some neurologists will set their low-pass filters there, lopping off the brain's higher frequencies to focus only on the most useful information. This is not to say that ECOG delivers no information in the higher frequency ranges, only that it's not always used clinically. But it was here, in these disregarded super reaches, that Moran and Luthard hoped to find neural gold. Coming from San Diego, Moran knew that individual neurons can fire well above 70 Hertz. Most critically, it is in these higher frequencies that neuroprosthetics have found the closest correlation between brain activity and physical actions. Could ECOG, unencumbered by low-pass filters, deliver the same high frequencies as penetrating microelectrodes? That was the question Moran and Luthard sought to answer when they went to check out the epilepsy monitoring unit. We look at the rack and I'm like, okay, why is this low-pass filter at 70 Hertz, recalled Moran. The theory suggested that there could be good activity higher up. Nevertheless, neurologists assured them that there was no usable information. Even Chalk, who had spent the last several years working in the lower frequencies of EEG, was skeptical. He came in and said, "Well, you guys listen to the bass. I want to listen to the treble." "Well, you guys listen to the bass. I want to listen to the treble," said Moran. Not being electrical engineers, they didn't realize they could also see higher frequencies. But I came from single units. It seemed obvious that there should be some power there. Still, it remained only a theory, and the pair waited anxiously as neurologists turned off the system's filters. Sure enough, a few seconds after they removed the filters, high-frequency gamma waves came dancing along the bank of computer monitors. Still, the real work lay ahead. They began each day unspooling a 100-foot Ethernet cable and taping it to the floor as they snaked the cord from the epilepsy monitoring unit to the patient's room. The cable quickly grew sticky from all the tape, but it allowed them to funnel the digital signals into the analog printer they'd set up in the patient's room. As the printer wheezed to life, it created an analog record of the brainwaves before feeding them into Luthart's digital converter, which squatted on a nearby cart. Luthart's converter redigitized the signal, sending them to a nearby PC that was running VCI 2000. Chalk software, in turn, ran the brainwaves through an algorithm, producing its results in the form of a cursor on a computer screen. Meanwhile, the research subject whose brain waves they were reading managed to control the cursor using only his thoughts. "It was amazing we saw anything at all," said Luthart. What they did see, however, was astounding. Luthart recruited 4 epilepsy patients as research subjects during that first trial, spending a few hours each day doing BCI experiments. They started small, using the same bulky electrode grids neurologists use to pinpoint seizure activity. The researchers asked their test subjects to perform 3 simple tasks: open and close a hand, stick out their tongue, and say the word "move." Ogeman had placed the electrodes over the area of the brain where the temporal, parietal, and frontal lobes meet, and as the research subjects performed their tests, scientists concentrated on the electrodes that register activity most closely correlated with the actions. In the study's second phase, they asked the subjects to imagine performing those same actions. Invoking a similar neural response, the researchers had programmed their computers to interpret increased brainwave activity as a command to move the cursor. If a research subject wanted, say, to move the cursor up, he thought of closing his right hand. By contrast, simply relaxing rather than thinking of relaxing, the right hand would move the cursor down. Whereas using an EEG to control a machine like this can take up to an hour of practice, the robust ECOG signals enabled their subjects to quickly gain control of the cursor. Moving randomly, a cursor would have had a 50% accuracy rate moving up and down. In his first ECOG run, however, the subjects accurately moved the cursor between 74 and 100% of the time, a success rate that researchers boasted that far exceeded EEG. It was the first time ECOG had been used to gain control of a computer, but what the young researchers discovered next turned out to be even more important. During the study, they asked their subjects to use a handheld joystick to move the cursor. By tracking the brain's high-frequency signals, Luthart and Moran soon realized that the certain frequencies were closely attuned to specific physical gestures. Move the cursor up. Neurons below a specific electrode would produce high-frequency waves. I didn't know if it was going to be as good as single units at that point. Single units were still the gold standard, said Moran. It turns out high-frequency brainwaves, stuff above 100 Hz, are basically an echo of single unit activity, so we can get the same information as single units. The experiments were more successful than any of them had dared to imagine. Moran was only just beginning his lab. He'd recently received 4 monkeys and funding to conduct single-unit experiments similar to what he'd done back in San Diego. But the ECOG experiment had him convinced as he worked through the first grants. He began using the implant chamber to record ECOG signals as well, essentially moonlighting to gather data for his next round of grants. After that first set of grants, where he piggyback. I never wrote another chronic single unit grant again. Meanwhile, Luthart was still in the midst of his residency. He was beginning to worry that their high-risk ECOG experiment was getting in the way of his safety experiment studying encapsulation in rodents. This was my meat and potatoes project, he said. I was really pained, embarrassed. I was like to Dan, oh, I haven't got much done on the rats. He was like, screw the rats. By then, Luthart was already thinking about intellectual property and inventing commercial products. He'd read a few books on the process and he'd written a patent for surgical retractor he'd invented. But he believed that they achieved with ECOG could be a game changer. I wrote the invention disclosures and tried to get patented right off the bat, he said. Donahue and his colleagues at Cyberkinetics had already implanted Nagel. Kennedy had his own private venture, Neural Signals, and Luthart was convinced that if neuroprosthetics were ever to become widely available, it would most likely be a commercial product. Let's say we did this and we gave it away for free. Thinking that we are doing a great thing, said Moran. If no company has exclusive rights, no company is going to build a product because they can't protect it. Intellectually property aside, the researchers saw another problem on the horizon. Market size. That's a fundamental practical reality, said Luthart. Even though there is all this great science going on, a venture capitalist isn't going to take a risk for a small market. And by that measure, the numbers just weren't there. The quadriplegic population of the United States is estimated at roughly 260,000, with about 12,000 new cases each year. Although there are an estimated 1.7 million amputees living in the United States, the vast majority of those are leg amputees and would not benefit from a neurally controlled prosthetic limb. It's not a growth market, said Luthart. You have to reconcile your noble intentions with the pragmatic economic reality of what the market will and will not support. How many new amputees are coming online each year? Maybe 11,000, 12,000? That's simply not enough for a venture capitalist. Stroke, on the other hand, is massive. Each year, more than 795,000 people in the United States suffer stroke. Roughly 130,000 people die annually, but for a population of roughly 7 million survivors, the results can be devastating. In the simplest terms, a stroke occurs when part of the brain dies after being cut off from its blood supply. Depending on where the cell death occurs, stroke victims can lose their ability to speak or understand spoken language. They can lose their capacity to recognize faces. They can even lose their sense of where their body is in space, leaving the patient mysteriously embodied. But the most prevalent complication by far is weakness or partial paralysis on one side of the body. The brain is wired for contralateral control, meaning that the left side of the brain controls the right side of the body. So a stroke that occurs in the left hemisphere can result in paralysis or weakness on the right side of the body. The country's largest stroke population would present investors with just sort of return needed to develop a commercial device. It wasn't immediately clear how Luthart and Moran might develop a neuroprosthetic for stroke. Nevertheless, they were convinced a solution would reveal itself, and Luthart moved to protect the new shiny data. We did that very early, he said. You make sure you have the science down, but first you make sure you have intellectual property that is protectable. As it turned out, just as a solution revealed itself a few years later when they discovered that neural features associated with movement were not confined to the contralateral sides of the brain. Using ECOG, they were able to decode movement features on the same or ipsilateral side as well, which in stroke victim would be the undamaged side of the brain. Would it be possible that they wondered to create a ECOG neuroprosthetic that tapped the ipsilated side of the brain to control movement? Their solution is the so-called IPSI hand, which they are developing through their startup Neurolutions, with an EEG headset linked to a motorized orthotic. The researchers come entrepreneurs hope recent stroke victims will use the device to learn to open and close the orthotic using signals from the ipsilated side of their brains. Their idea is that by thinking about moving the damaged hand while wearing the neuroprosthetic, a patient will eventually rewire his brain, training his ipsilateral side to open and close the biological hand. An implant would give him better control, but that would entail a lengthy and expensive FDA approval process that a small startup like Neurolutions couldn't afford. The IPSI hand is really a beachhead to show that BCI is clinically relevant, said Luthart. We can do a better job with an implant, but EEG is a low-risk entry. What they really need, though, are good results and investor dollars, and they hope the EEG-based IPSI hand will convince venture capitalists that it's a viable product. There's a real believability gap. It almost seems like science fiction, he said. But as people become comfortable with its clinical application, it really opens the door for believability for doing an implant. The company is now raising investor capital and conducting a small clinical trial where participants use the device to achieve 1 degree of freedom, opening and closing the damaged hand. Neurolutions still has a long way to go, but if the IPSI hand is successful, with Hart Moran and their cohort stand to benefit greatly. Their idea of using ECOG for BCI is now known as patent 7120486B2, which names them as the sole inventors of any brain-computer interface that employs an electrode grid implanted beneath the scalp to collect ECOG signals from the wearer. Any BCI that uses ECOG, that's us, Luthart said one evening while seated at his kitchen counter and scrolling through a list of his intellectual property. Between the surface of the brain and the scalp, that is what we own. Basically, if you try to decode invention of ECOG BCI that's covered by this patent, it's very, very potent claim. Chapter 6, The Backup Plan. Andrew Schwartz knew that if he wanted to stay relevant, he needed to sink his penetrating electrodes into the human cortex. DARPA could provide that opportunity, but the agency had opted to go with the Applied Physics Laboratory at Johns Hopkins. They have tons and tons of military contracts, so they're used to dealing with these guys, he said. They have a comfort, and they could do all of these 3D Gantt charts, which DARPA seemed to like. When DARPA announces a project like revolutionizing prosthetics, it also releases a list of potential performers. Research laboratories like the agency is willing to fund as part of the project. Any researcher or lab that competes to administer a project can choose from that list. Building a team across institutions for Schwartz, that meant working with a project manager and a select group of robotics experts to build an arm before it linking to the brain. There are less than 6 people in the world that really know how to build a robotic arm, and they all come from MIT, said Schwartz. All these other yahoos basically said, oh, we can build a robotic arm. We know that what we're doing. He added that both the Hopkins team and Dean Kamen's team talked to him about joining their DARPA proposals. It's like I'm sitting there, you're going to be my boss, he said. Needless to say, I didn't get on any of the teams. Schwartz was effectively locked out. The Pentagon had shut the door to humans for him, but DARPA funders were far from cutting him off. They wanted him to keep working with monkeys and awarded him $2 million for a study that would not only catapult Schwartz's research onto 60 Minutes and into the pages of New York Times, but would eventually give him a shot at the human motor cortex. They had people doing the same kind of thing that I was doing. A lot more people with a lot more money, and they didn't get anywhere. He said, they kind of kept me as a backup plan. Other researchers were circling around the problem of how to link the brain and multi-jointed prosthetic limb, but few researchers had successfully closed the loop with the robot arm. Earlier closed-loop work had taken place either in the virtual environment of a computer screen or at a safe distance, as with Matthew Nagel, who performed a simple pinching action with a prosthetic hand. Mental control of a cursor would be a boon of quadriplegics, but DARPA wanted a brain-controlled prosthetic limbs you could use to brush your teeth or comb your hair. The race was on, and Schwartz devoted his research funds to a suite of experiments aimed to directly at DARPA's goal, an elegant neural control of dexterous multi-jointed limb. It turned out to be great, he said. I didn't have to report an APL or anybody. I just did my own work. With the electrodes in hand, Schwartz and his colleagues began work with 2 monkeys and a pair of robot arms, outfitted with pincer claw instead of a hand. Training research monkeys falls somewhere between art and science. Since you can't tell a monkey what to do, researchers must devise ingenious ways of familiarizing the animals with the physical essence of a task. It's a delicate procedure, and Schwartz began by training his monkeys to control the arms using a joystick. Pressing the joystick forward, the animals learned they could extend the limb to various fixed points in space, grab a marshmallow or a sliced grape from a skewer, and pull back on the joystick to bring it to their mouth. As the monkeys brought the marshmallow back, researchers impaled the next food reward, fixing it in one of the 4 positions for the animal to grab. Once the monkeys were familiarized with the task, researchers removed the joystick, immobilizing the animals' arms by placing them in tubes and attached to chairs. Meanwhile, they recorded neural activity while placing the arm under automatic control, giving researchers command over the arm as it grabbed food and brought it to the monkey's mouth. One of the great discoveries of the late 20th century happened in the lab of the Italian neurophysiologist Giacomo Rizzolatti. The scientist had implanted electrodes in the premotor cortices of monkeys, hoping to listen in on neurons he believed were associated with hand and mouth movements. The researchers recorded from individual neurons as monkeys researched for peanut, tracing the cell's firing pattern before, during, and after the movement. By the measure, Rizzolatti's experiment did not differ tremendously from neural recordings his fellow researchers were making in other labs. What set Rizzolatti's work apart, however, occurred by accident. During a break between tasks, the monkey sat idly in its chairs as researchers milled about the room. The monkey wasn't moving at all, but when one of the researchers snatched a spare peanut and popped it into his mouth, the neuron had been recording erupted as if the monkey had grabbed the peanut itself. It was a shocking discovery. The brain, or at least a specific class of cells, seemed not to distinguish between an action performed and an action observed. Here was a class of neurons, later dubbed mirror neurons, that was involved in motor planning, but that was also interested in physical actions of others. Much has been written about mirror neurons. The brain researchers, such as Marco Iccaboni at the University of California, Los Angeles, have proposed that the mirror neuron system plays a critical role in recognizing the needs of others. We flinch when we see someone injured on the street. We thrill at most any sport, and we feel deep sympathy for the fictional trials of characters in film and theater. Why? Because at some level, our brain physically recreates the experience as though we were our own. Mirror neurons, these researchers believe, not only are the fundamental mechanisms by which we feel empathy, but also play a role in so-called theory of mind, enabling us to recognize that other people have ideas and desires that are distinct from our own. At our more practical level, the brain's penchant to recreate observed action helps researchers such as Schwartz to prepare the monkey's brain for BCIs as the monkey watched the arm grab a piece of food. Animals' motor neurons began firing as though They were grabbing the fruit with his own biological arm. Meanwhile, Schwartz and his colleagues used the information to build their decoder, the computer algorithm that associates specific neural firing patterns with particular movements. As the researchers continued to move the arm, the algorithmic association between firing patterns and the robot arm movements grew stronger. Eventually, they began to dial down their control of the arm, blending automatic control with signals from the animal's motor cortex. The monkey had partial command of the arm, but scientists could still correct its movement. If the arm began to go wildly off course, they had effectively given the monkey training wheels, encouraging it to move the arm in desired back and forth directions, but constricting the arm's movement from side to side. It was a synergy between animal and algorithm. The computer is learning to better interpret the monkey's neural code. The monkey's motor neurons were learning to better control the arm. Earlier centered out experiments gave animals fixed beginning and end points to their task. The monkeys knew to begin the task when the cursor appeared in the center of the monitor. Once the animal successfully moves the cursor to a target, it would receive its reward and task would reset. The animals needed only to control the cursor for the time it took to move it from the starting position to an end target, sidestepping the added complexity of controlling the cursor for long periods of time as it traveled home. Schwartz, by contrast, gave his monkeys continuous control of the arm. Unlike earlier studies, the animal didn't return to a home position after grabbing and eating a food reward. Rather, the monkey had to maintain control of the limb as it reached to a new location for the next reward. It was a complicated task, but again, Donahue had beaten Schwartz to the punch. Using the BrainGate system, Matt Nagel had not only managed to pinching action with a prosthetic hand, but also gained continuous control over a computer cursor, using it to navigate a desktop environment. Opening email and playing music, he could hardly move that cursor on the screen. It was terrible, Schwartz said of Nagel's shaky performance. They point to my stuff, they said. Well, you can get it to work in monkeys, but it won't work in a human. Donahue's performance might have been unconvincing to Schwartz, but there was no denying that he'd granted a human research subject continuous control of a computer system. What Schwartz research lacked in novelty, however, it made up for in sophistication. Using his brain-computer interface, Schwartz's monkey gained 4 degrees of freedom to perform an essential function, self-feeding. Perhaps even more important were the behaviors that began to emerge. Rather than using a neural cursor which exists only in virtual realm, Schwartz monkeys were directly interacting with a physical limb. In a video of the research, one monkey sits in a task chair, the view of its protective resin cap obscured by aluminum scaffolding. The monkey's arms are immobilized in plastic tubes. The black ribbon wire that emerges from the skull, it peeks from behind scaffolding whenever the animal strains its neck. Meanwhile, the industrial-looking robot arm, a device of shiny steel tubing and pincer wrapped in white fabric, methodically reaches to grab marshmallows. The monkey flexes its right arm as the robot reaches for a marshmallow. Bringing the food to its mouth, the animal strains towards the marshmallow, successfully completing the task several times in a row. Researchers sampled the monkey's brain signals every 30 milliseconds, giving them about 33 snapshots per second of the monkey's neural activity. It took the algorithm and robot arm 150 milliseconds to transform each neural impulse into an arm command, which is about the lag time of biologically intact nervous system. In essence, Schwartz and his colleagues had given the animals instantaneous and uninterrupted control of the arm. Nowhere was this more apparent than in the second video when a researcher moves a piece of food away as the monkey in the mid-reach. The animal quickly changes course, grabs the marshmallow, and takes a bite. In a third video, the monkey partially takes the marshmallow into his mouth. With the biological arms restrained, it holds the marshmallow in its lips, using the pincer to push the treat all the way into his mouth. It wasn't making rote, stereotypical movements. It was freestyling. Of course, biological intact person continually adjusts their movements to better adapt to the shifting environment. By that measure, the monkey's movements are banal. But that's really the point, a smooth, naturalistic movement that approaches its biological analog. The monkey was coming to embody the robot arm, as its neurons spontaneously adjusted to better achieve the task at hand. At that moment, Schwartz's monkey were doing something not so different from what a baby does when she learns to walk or when a child learns to throw a ball. But while biological coordination improves as pathways between neurons are streamlined and strengthened, the neurons are here changing their behavior to better control the non-biological arm. They were adapting to the interface.